an honor since a few times of these meetings to be with you and to help out a little bit as well. So my first speaker today is Dr. Jung Kim from Korea. And so Jung got a PhD at Amherst in '95, and then was with Khorana, I've been told, from '95 to 2003. And I met him there, and those were the times in Gobin's lab where the dark room took a central place in the lab because people were working on these light-sensitive receptors. And now I've just seen that Jung is working on another dark room altogether, which is called the ocean, and so we'll hear about that.
It's really my pleasure to be here in India. Actually, I was trained by two Indian faculties. One, Shilda Sarma, who was my PhD mentor in University of Massachusetts. And then I moved to Gobin's lab for quite a while. And in addition to that, in our family, we believe that our great-great-grandmother came from India, and then to start our family degree in Kim's family. Okay. What I would like to introduce you today is photoreceptors in fishery system and photoresponse. I would like to start my talk with a brief introduction about rhodopsin and light-emitting diode, which I used for examining the photoresponse. And then I will go to the spectral characteristics of fishery rhodopsins, and then I will talk about the construction of LED modules and behavioral monitoring system and photoresponse analysis. And vision is mediated by absorption of light by photoreceptor in our eyes. There are two kinds of photoreceptor cells in vertebrate. One, rod cell, is responsible for dim light vision, and cone cell is responsible for bright light and color vision. And rhodopsin is a photoreceptor responsible for dim light vision, consisting of protein, opsin, and 11-cis retinal. And it has been used as a model system for studying G-protein coupled receptor, which is one of the most important target for drug discovery. Rhodopsin has been used as a model system for studying G-protein coupled receptor, mainly due to it is easy to prepare and easy to analyze the structure and function of rhodopsin. For example, if you take just UV visible spectra, then you can tell whether it is in the inactive state or in the active state. And it is also important to study the visual signal transduction and structure function of G-protein coupled receptor. The reason why we are interested in fishery rhodopsin is aquatic environment is slightly different from the light environment in terrestrial system. Because in terrestrial system, there are multiple wavelengths visible light is present. But if you go to the aquatic environment, depending on water depth, you will face the different wavelengths of light. This is due to differences in light absorption of the water, which we call spectral filtering of the water. So as you can see here, for the light with longer wavelengths can be observed by the water from the surface. But for the light with shorter wavelengths can penetrate further down to the bottom. So the light environment we can expect is probably up to 200 meters in clean water. We expect to see the blue light, but we don't expect to see the long wavelengths at the bottom. So since the light environment is different, depending on water depth, probably fishes living under different water depths may have different the light harvesting system. And so for, if we study the fish with habitat adapted photoreceptor, then we can provide some answers for the spectral tuning, fine spectral tuning of rhodopsin and G-protein coupled receptor. We can also get some information for the evolutionary study of the fish. And the topic that I'm going to talk today is related with fishing light. The system to examine the effect of light was light emitting diode. Light emitting diode is the diode converting electrical energy to the light energy. It is known to be efficient in terms of the energy production, and it is more durable, and it is eco-friendly as compared to the traditional light source. So you can see the many applications of LED, from lighting to display, and you can also see the application to the medical and biological science, even in agriculture and photobioreactor, and the topic of today's discussion, fishing lamp. And the reason why we are interested in LED technology in fishery is that so far people used the light source from the traditional, the incandescent light lamp or methyl halide, but it is known that these are the less efficient as compared to the LED light source. So the government in Asian country were trying to replace the less efficient conventional fishing lamp source to the more efficient LED light source. And from the initial stage of application, what they noticed was that it can save energy a lot. But there was some concern on catching efficiency. For some types of LED, it is less efficient than operation using the conventional light source. So what we thought was probably this may result from differences in the characteristics of light source between traditional light source and LED light source. As you can see here, the daylight and incandescent light covers wide range of visible spectrum. However, the difference of LED different from the traditional light source is that they emit the narrow spectrum, narrow range wavelength spectrum. So this is the main difference between two light generating systems. So during the first stage of the fishing lamp, the replacement, what people tried to do was just to incorporate the blue LED to the system, which showed the deepest penetration. But since there are some concerns on catching efficiency, in order to solve that kind of problem, we need to understand the biological characteristic of target fish's visual system. This is the main reason why we started this. So what I'm going to show you today is to examine the absorption characteristics of photoreceptor and then compare to the effect of LEDs of different wavelengths. What we did was we examined the photoreceptors in one of the fish, chub mackerel, which is one of the species caught by the night light fishing. And another fish I'm going to show you is rock bream, which is common species produced by aquaculture in Korea. And what we did was isolate the rod out segment from these two species, examine the spectral characteristic of the photoreceptor, and then illuminate the LEDs with different wavelengths, and then examine the photoresponse and photosensitivity of the receptor. So this is one of the results. The photoreceptor analysis can be done like in bovine rhodopsin, which we did in Govin's lab. The one thing we can do is to express opsin and to regenerate rhodopsin, and then we can examine the spectral characteristic. Another way we can do is to purify the isolate rod out segment and then check the spectral characteristics. So this is one of the results. Just isolate the opsin gene from chub mackerel and by PCR and try to express in animal cell system like bovine opsin we did. And then the left panel shows the expression of the mackerel opsin in the right lane. This result shows we can detect the expression of mackerel opsin, although the level is much lower than bovine opsin. This may result from some differences in the sequence. And then we reconstitute rhodopsin and then check the absorption maxima by taking UV visible spectra in the dark and after the light activation. So we can obtain the different spectra, which we can subtract light spectra from the dark spectra. So the result you can see is here is the mackerel opsin showed the absorption maxima around 500 nanometer. And then we also checked the spectral characteristic of fish by rod out segment preparation. Before we do that, we can check the rod cell and cone cell by histological analysis. And then we isolate the ROS. We did ROS preparation. This is the result. I can show you the spectra taken in the dark and after the light activation. And you can see the absorption maxima around 500 nanometer region. So we can confirm the absorption maxima of rock bream, the ROS, which is around 500 nanometer. If you are in rhodopsin field, and if Govin is here, I don't think he likes this kind of spectra at all, because there are too much peak at 200 nanometer. So in connection with this, I put one slide, but if one of the assignment for the people who came to Govin's lab in rhodopsin age was, first assignment was we were given a vial of a vial of the bovine retina, which contains 50 retinas. And then he asked us to isolate the rod out segment from there. And the reason for the asking that is he asked us to be accustomed with protein purification, especially in the dark room. We should do all the preparation in the dark room. And as far as I know, the people who came in 10 years adoption period, everybody did it, those experiments. Then Govin knows what kind of yield he expects. It usually 45, between 40 and 50 milligram out of 50 retina. And then he also expects, I put one of slide, but it is somehow missing. As soon as he saw the spectra, then if he saw this one, usually if you have really purified the adoption, then the ratio of 200 and 500 is between 1.7 and 1.8. So immediately, if you show him this kind of data, then you are kind of in a cautionary. You should be in a cautionary mood. And you should be careful for the next experiment. You should be very meticulous for carrying out the next experiment. And another thing is you can also see the 500 nanometer. So if he usually asks us to show the illusion profile and spectra in the dark and spectra after the light activation, then he can compare my result with the result in his brain from the old experience. So if you show him a good result, then you will probably okay for 2 to 3 months. But if there is some kind of the 380 nanometer peak in the dark spectra, then he believes you are kind of person who is not meticulous enough to do the experiment in the dark room, like that. Anyway, from this experiment, he asked us to be accustomed to carry out the experiment in the dark room. And then, in order to examine the effect of light of specific wavelengths, we constructed the light LED module system. And with the help from LED Marine Biology Conversion Center in our school, we made the custom design LED system with different array and different size. And then this is the module LED system we constructed with different wavelengths. For example, here, we covered the wavelengths from 405 to 660 with the more or less similar interval. So we can examine the effect of specific wavelengths from this by using this. And after constructing the LED module system, we need to check the efficiency of LED modules by using a radiometer. And we can confirm from the spectral power distribution curve, by using radiometer, we can confirm the peak wavelengths and light intensity of the LED modules we constructed. And next one shows another set of LED module we constructed. And by adjusting the voltage and current, so we can set up the system with LED of different wavelength, but with the same photon intensity. So then we can compare the effect of specific wavelengths by using this system. And after constructing the LED module, we use two sets of systems to monitor the fish behavior. One is by using underwater acoustic camera system, and then the another system we used was video tracking system. And this is the picture of the acoustic camera system we used. And then we set up the system in a fish tank with the 3-meter diameter, and then put the 20 fishes, 20 mackerels, chopped mackerels in the tank. And then we acclimate the fish in the dark, and then illuminate with different wavelengths LED system. These are the system, LED system we set up for the experiment. And the experiment, the way we did this experiment was this. First, we acclimate the fish in the dark, and then illuminate with different wavelengths. So this is the result you can see. So by using the acoustic camera system, we can monitor the moving of the fish under different light conditions. So the way we check the photo response is by checking the speed and frequency of fishes upon LED different light conditions. So this is the result I can show you. This is different, the experiment condition, for example, from dark to red, dark to cyan, dark to blue, and different the LED. This is control experiment with white LED system with the same photon the amount. And what you can see here is this is the rate increased by LED illumination. So what you can see here is upon illumination of cyan LED, it shows the most drastic change as compared to the others. So what this means is that cyan is more effective to induce the photo response in fish. And this is second set of experiment. Since we notice the red light has few effect, almost no effect on inducing the movement. So what we did, the next step was, first we acclimate the fish in the dark, and then we acclimate in the red LED light. And then we further, for the second stimulation, we use different wavelength LEDs, whether we can see the same type of stimulation or not. So this again shows the changing from red to cyan shows the more drastic changes as compared to the others. This again shows the more effective, the induction effect by cyan LED, which shows the peak, the wavelengths, the consistent to the absorption maxima of the ROS. And this is the summary of the result from the photo response analysis. And as you can see, cyan is more effective in inducing the movement as compared to other LEDs. And the other system, the monitoring system we used was video tracking system. Basically, for the fishes which move slowly, we can use, it's okay to use this video tracking system. So in this system, we put fish and then track the movement of fishes, the experiment fishes in the tank, in the dark, and upon light activation. We also, for the other fishes, we have the Y-shape, the experimental system, for example, to put to put the target, the animals here, and then illuminate from the side. And then whether it can go toward the light source or avoid the light direction. This is another system we constructed. And this is the result from video tracking system. As you can see, it's similar to the previous result. Cyan is more effective in inducing the photo response in fish. And in order to examine whether the photo response in fish result from the absorption maxima, whether it is related with absorption maxima of the photoreceptor in fish. What we did in this experiment was we have ROS purified and then illuminate the ROS, the preparation with red LED and different LED system. And then we examine the level of decrease in the level of photoreceptor. What you can see here is again the cyan is more effective in photosensitivity reaction. So as compared to the others. So overall, what we can see here is the effect of the effect on photosensitivity and photoresponse result indicates that the 505 nanometer LED is more effective than the LED with different wavelengths. And this is same the result we can see in photoresponse and the photosensitivity of ROS preparation. So this is a summary of the result. So we examine the absorption maximum of the ROS in fish, which turned out to be around 500 nanometer. And we examine the effect of LED of different wavelengths. And then the conclusion we have is light with wavelengths close to the absorption maxima of photoreceptor is more effective than the other LED, of which the wavelength is distant from the absorption maxima of the photoreceptor. And this may be applicable to the plant system. One of the application could be if you want to grow the plant by using LED, then you may better to check the absorption spectrum of the chlorophyll and in the plant. So then you may have better the efficiency in growing the plant. And this is all I have for the research, but I would like to share a few slides of COVID. And this is the photo that I took from Japan meeting. And this is the photo that I took in 2007 in Govin's place in Cambridge, when I visited the Cambridge. And this is same photo the Joydeep showed. Now you know who took this picture. So I have this photo, but I am not in the photo. So you may guess. And this is the last one. And this is the kind of preparation that I got when I left the lab, and in 2003. So as you can see, we always, like other group photo, we always have the same group meeting with Tom's group. And you can see our great mentor, Govin and Tom. And you can recognize Judy. And at this time, the people in Govin's lab was the Phil Reeves and Parvati, Prasen, who is going to give a next talk, and Yume. And this means I am the fifth person from the last in the lab. It was a really great moment to be in Govin's lab, and it was also really great experience to learn from Tom. So it was really good experience for me. And this is in collaboration with LED Marine Biology Convergence Technology Research Center, and Junchul did most of the work for this experiment. Thank you.
Thank you, Young. There's a question over there.
Hello. How did you plan that experiment that irradiating the fishes with different wavelength will actually result into increasing their mobility? Like, what is happening at the level of the fish? I mean, why their mobility is increasing? I mean, how you got the idea of designing that experiment?
So, idea to examine the effect of different wavelengths.
Yeah, so on fishes. I mean, what is happening with the fishes that they are like, just, randomizing with some speed? So
the difference we checked in this system was we measured the speed of fish moving in the dark, and then upon light activation. So if there is no effect, so then speed should be the same. But if there is an effect on inducing the photo response, then there must be change in the speed of fish. Or you saw the kind of spectrum in the video file. So another way we can calculate the movement of fish is that we can calculate the frequency of fish appear in that section. Okay. So that's the way we can check.
All right. So it is more about the visibility, you are saying? Or
so in one of the applications for fishing lamp is rather than just incorporating the blue light LED, which show the deepest penetration, it may be better to include, to induce more efficient movement. It may be better to include the LED of wavelength, which is consistent to the absorption maximum of the photoreceptor. That is more effective in inducing the response.
All right, thank you. I think we have to move on.
Thank you.
Thank you. So we'll continue this session on GPCRs. We'll continue the session with Dr. Prashen Shelikani. He had a PhD of the University of Manitoba, which is in Canada, and then associate with Govin's lab from 2003 till 2007. And then he moved back to Manitoba and rose through the ranks of the professorship and is now a full professor at that university. And so Prashen will discuss taste receptors, in particular bitter receptors, for us today. So Prashen, please.
Thanks, Nico. And thanks to Tom and Rajendra for organizing a wonderful symposium. Definitely, yeah, this is one of those, keeps on going, eh? That's nice. So, as you can see, I am in the Department of Oral Biology. I think most of you don't know what oral biology means, right? So as I go through the lecture, I'll be talking about taste receptors, which makes more sense to be in oral biology, right? Okay, so before I move on with the science, actual science, this is something we do other than science, used to do other than science in Govin's lab, eh? And Tom was the skipper. And so we went, well, this has nothing to do with Jong's thing, eh? Well, it's more of a coincidence, the deep sea fishing. So deep sea fishing means, yeah, we used to go 30 or 40 kilometers, I think, each day. So the day starts at 5 in the morning, and half a day or full day? Full day. So we went for 3 years, at least when I was there 3 years, and the irony is I never caught a fish, eh? So when I used to come back, my wife used to say, okay, where is the fish? Eh? She was a little bit, first year it was okay, after that it's like if you're not showing the fish, eh? Like it's, anyway, so that was like one of those, it's very, very interesting. And then these are pictures from Gobind and Tom's lab from 2004. So you can see the Whitehead Institute in the background, Tom and Gobind. Here they are sort of towards the end, eh? So they are sort of flanking the group, eh? And then, you have Phil, a lot of people there, Judy, Parvati, and you can see me at the back, and Jay, Phil, a lot of people, and you can see Gobind in action, sort of moving around, very active. And that's our building, eh? The biology building in the back, that's where the lab used to be. And 2006, fast forwarded, this was, I think, the last Christmas dinner we had as a lab. So that was Gobind, that's Julia, Gobind's daughter, Mike, Judy, and Parvati. She was a postdoc along with me. So we were two postdocs towards the end. And so she's now a lawyer, eh? So she felt law is better than science, so that's good. So that was our, these are some of the memories from the 4 years, close to 4 years I had with Gobind. He is more than a person for me, so. Moving on to what I did after that, 2007 is when I started my research. So the first couple of years, I had to figure out what to do. And one of the things is the acting dean at that time, when I was recruited, he said, okay, you're working on GPCRs. How does that relate to dentistry? So I had to look for something that is orally relevant. And that's when I figured out taste receptors. So it's more by chance that I happened to actually work on taste receptors. And these are GPCRs, right? So right now in my lab, I have a couple of different areas, like we have basic and clinical studies looking at structure, function, and physiological role of these bitter taste receptors. And then we also have applied industrial collaborations, grants, and startup companies that are involved in taste blockers, elucidating new blockers, and also development of GPCR autoimmune diagnostics using GPCR autoantibodies that are present. So this is in collaboration with a company from New York. Moving on to the basic science, like that's what I'm going to mention today. Human taste perceptions. These are all considered as taste perceptions, right? But unfortunately, hot is not an actual basic taste. Hot means we are sensing pain, nociception, right? Same thing with cool, menthol. It's not considered as a basic taste. So people always get confused between hot, cool, spicy. Spicy is not an actual taste. It's a combination of tastes, right? So what are the basic taste sensations? There are 5 of them so far. Things might add on down the road. Of these 5 taste sensations, umami, sweet, and bitter, these are the ones that are sensed by G protein coupled receptors, or GPCRs, right? And of these 3, umami and sweet are sensed by 3 receptors, depending on how the receptors combine. So you look at the green one here, that is common for both umami and sweet. That is known as the T1R3. And then you have the T1R1 that is specific for umami. So this thing, when it combines with the green, the T1R3, it gives you the umami sensation. The same thing with sweet, right? And when you come to bitter, so these 2 belong to class C GPCRs. GPCRs, it's a big family. And these 2 belong to the class C, the metabotrophic receptors. Whereas bitter receptors, there are 25 in humans. And right now, they are classified as class T. That's a recent classification. No basis for that. So they're sort of an orphan. They float around the GPCR family, but there are 25 of these. So these receptors constitute the 3rd largest among the GPCR family, if we can actually look at it in a different perspective from the chemosensory angle. And coming to the other taste sensations, the salt and sour, these are sensed by channels. So my topic is on bitter receptors. As I said, there are 25 receptors in humans. And these things are not just present in the oral cavity. They're present throughout the system, right? And we don't know what they're doing in different parts of the body, right? And one of the things we are focusing right now is cystic fibrosis. And I will be looking, we are pursuing certain angles in that, which I'm going to talk in the next few minutes. So, cystic fibrosis, what is that? It's a genetic disease, an autosomal recessive mutation. CFTR, so here you're looking at healthy chloride transport. It's a transport mutation, the CFTR, which is a transport channel. Things normally go okay in the healthy state. In the disease state, you don't have efficient chloride transport. Because of that, sodium gets transported in, but chloride is not pumped out. Because of that, you get a thicky mucus. And that mucus acts as sort of the nutrients for bacteria to grow on. That's how bacterial infection starts. And that's the leading cause of death. So bacterial infection leads to chronic inflammation, and that leads to failure of the lung. That's the sort of the pathway that occurs. And according to Cystic Fibrosis Canada, CF Canada, the median age of survival in Canada is around 48 years. The disease is not curable. It's not just confined to North America or Europe. This is also present in China, India, but it's not detected. That's one of the things that is coming up now. And whether it causes the same phenotype, that we don't know yet. The cost per patient is around 300 to 400,000 in Canada, because we have a global health supported system. The government supports the health care, so the costs are pretty high. And these are totally covered by the government. So it is an expensive to treat this particular thing. So as I said, CF is predominantly a bacterial infection. Bacterial infection is the one that causes inflammation and then lung failure. So how do these bacteria talk to each other? Most of us know, quorum sensing, right? Depending on which molecules are synthesized. So each bacteria have their own sort of molecules that they synthesize. For example, here we are looking at HSL, homoserolactones, and those. And Pseudomonas aeruginosa, that's the main culprit here in CF. That's the one that causes infections in CF. There are a lot of other bacteria too, gram-negative and so on. But so these are some of the QSMs we are looking at. And also the antibiotics that are used in CF treatment, levofloxacin, tobramycin, azithromycin. The reason why I'm mentioning these three, we'll be looking at these in the next few slides. This received approval from Health Canada in 2016. That's last December. So it's now publicly available in Canada, not in US. It's available in Europe and Canada. So again, we'll be looking close into these three antibiotics. So, coming to the relationship between T2Rs and quorum sensing. The first study that showed was published in 2010, where they suggested that quorum sensing molecules, AHLs, along with T2R agonists, can activate the airway cells through T2Rs. So that was one of the first studies that showed. After that, there were many studies in JCA and many other journals, including chronic rhinosinusitis. That's a condition, inflammation of the sinus, where things, bacteria play a major role. So T2Rs are known to be involved in interacting with QSMs. But what's not known is how does it play in a CF condition, right? And also what's not known is how do these molecules taste. Getting a human taste panel to taste these molecules is tough. Getting ethics approval will be beyond it's right. So to go around that, we use something known as an e-tongue, an electronic tongue. That's what you're looking at here. It's like a pH meter, right? It has all these sensors. And those are the sensors that dip into these things, unknown solutions. It gives a readout. The only issue here is it's $200,000, pretty expensive, right? And so what does it do? You can take, so here looking at a table that it generates, like the first few compounds are better compounds, caffeine, quinine, dextromethorphan. That's a cough suppressant, right? And these are the concentrations that are used in these carousels, in these vials. These bitterness scores are from an actual taste panel. So for caffeine and quinine, it's easier to get an ethics approval for a taste panel. So those can be done. So those are the actual results from a taste panel, whereas these are the predicted scores from the e-tongue. So all this column represents the score from the e-tongue. So if you look at these, it's pretty much close, sort of agrees with that. So dextromethorphan, that's not known. So that's what it says. So the higher the score, the more better it is, right? So these ones, the ones you're looking towards then, these are the quorum sensing molecules. So we use them as a load because in CF, it's not just one quorum sensing molecule. It's a combination of them that causes the infection, right? So in CF, the load is high, right? So we did, the load means we are combining a couple of these molecules together and then looking at it, right? And if you look at these numbers, look at these low numbers, 0.05 millimolar, and you're looking at 12 plus. It's more than what quinine or caffeine tastes like. So the taste of these, the bitterness of these, according to the E-tongue, these are very, very bitter. Right? So now the actual thing is, okay, the E-tongue is saying something. Does it actually translate into a response, a physiological response? So that's what we are trying. So this is the hypothesis. Like these are the pathways that T2Rs act through. We know bacteria act through the LPS and TLRs. That's already a well-established pathway. But what's not known is whether these things act through T2Rs causing inflammation in the CF airway. So what we did is we used encounter analysis, sequencing. So we sequenced a lot of tissues, not only from patients. These are CFI is cystic fibrosis established cell line, immortalized. NULI is an airway cell line, immortalized. And in addition to these, we also looked at breast cancer cells, MB231, MDA, MCF10A, and a lot of things. But what we found is the expression pattern is similar. These 25 receptors, irrespective of which cell line you're looking at, the way they are coming up is similar. Right? And again, it shows like here, unfortunately, that's one of the disadvantages with encounter. If the sequence homology is very high, more than 90%, it's very tough to design probes that can detect those between the different genes. Right? So we did validate some of these with the flow cytometry. Getting antibodies is also tough for these, but for the ones we had, we looked at those and it is. So as I said, there is a specific pattern of expression with 14 and 20, the one that are highlighted in blue, showing the highest irrespective of which tissue we're looking at. So both in CUFI and then CF and the normal, it's the same. Eh? That means there's no differential expression based on the disease state. So we also looked at donors, CF. So this is before and after lung transplant. Bronchoscope is from a CF patient. Eh? We had up to 5 donors, so depending on the heterozygosity of those people. So, and again, that's to take a what we saw with the primary cells from the donors actually applies to these stable cells. And then we wanted to figure out which quorum sensing molecule is actually involved. So we looked at C4, C8, C12. C12 is one of the most common ones. So we looked at those with respect to each of the T2Rs in stable cells. Eh? And what we found is C12 activates 3 of the 4 we tested. Eh? And some of them, they don't, like T2R1 didn't get activated. So there is some diversity here, but the trend looks similar. Eh? So we are also looking at the physiological effects, which I'm not going to go into. What happens, how do they cause inflammation? And those responses, those are the ones we are looking at in that. But what's more interesting is the antibiotics. And here, what we already know is that, okay, these things kill, right? And what's not known is, what is the effect of these antibiotics on these? Because remember, these are mostly bitter tasting drugs, right? And as I said, levofloxacin was released in Canada in 2016. It's the first fluoroquinolone antibiotic to receive marketing authorization. Eh? So around the same time, that's last December, we got an inquiry from this company. They posed these 3 questions to us. Eh? So the first thing is inhalation of an antibiotic with a very bitter taste, such as levofloxacin. Could it have any additional effects? Additional means non-antibiotic effects, right? So they wanted to know what else it's doing other than killing bacteria, right? And also they are looking at, is there any possibility to define the difference between the effect of T2Rs between different inhaled antibiotics? So it's not just levofloxacin, they are also interested in other things. Anyway, we didn't get funding from them. Things change fast with pharmaceutical companies. This particular division from Horizon Pharma, the CF division, was sold to Chelsea, Europe, and those guys just fired all the CF people there. Right? So it went pretty fast. But anyhow, so the things that they posed were very interesting. So we started pursuing the first two. The third one is more of a physiological impact there. So the first is to define whether these antibiotics activate T2Rs. If so, how many, and what is the specificity? So again, we use the same thing, E-tongue, right? So we looked at these, and these are the scores. So some of them are very bitter, like azithromycin, looking at a score of 15, whereas levofloxacin, it's around 4. Levofloxacin and tobramycin, those are the two commonly used CF antibiotics in CF treatment. Those are. And what's interesting is the ones that are highlighted, the one in red, levofloxacin and quinine, they share the same quinoline ring. So, so we wanted to test out, okay, this one is highly bitter, even at 0.03 millimolar, whereas this is, even if you take 1 millimolar, it's not showing that. So does it correlate to the number of receptors it is activating, bitter receptors, or is there something else at play? Right? At the same time, we are looking at between the different cell lines. It can be just error prone too, right? So we wanted to test it out, do a docking, so that's what we did here, a pharmacological characterization. 8 of the 25, whereas levofloxacin, so far we found it activates 3 of the 4 we tested. So there are structural variations there which can be explained. And then we also looked at 14, that's the other one. Flofenomic acid, it's sort of a cognate ligand. I couldn't say, I can't say it's a cognate because it's a synthetic one. But that's the one, that's an agonist, just like quinine for T2R4. Flofenomic acid activates T2, is known to activate 14. So the same thing with diphenhydramine. That's also known to activate T2R14. Tobramycin, levo, the same 2 things we tested. Again, it's a mutational study. Take-home message again is ECL2 plays a critical role. There are few residues. The moment you take them out, the receptor doesn't bind, right? Whether irrespective of which ligand you're looking at. So that's, again, not known in the field. This is something new in the taste field. So this is where we are at, like I'm not going into the physiological role. We're still looking at what happens in an actual CF patient with these. So coming to the conclusions. First one is no significant difference in expression between T2Rs, in T2Rs between CF and non-CF cells. And these quorum sensing molecules, along with the bitter tasting antibiotics, can activate T2Rs. But the way they're doing is different. The mechanisms might be different. So the first question, to answer one of the first questions from these guys, yes, they do have predominant roles. Antibiotics can have non-antibiotic roles, depending on the tissue they're acting on. And then we are also looking at, as the third angle is the physiological effect of these, the QSMs in CF. That's something we are looking at, the inflammatory cascade. Our hypothesis is they are anti-inflammatory, and we have data that shows IL-10 and certain other cytokines are elevated upon activation by these QSMs. And also not only in CF, we also have it in the oral cavity. Strep mutans secretes CSPs, competent stimulating peptides. And we have data that shows that those things also activate T2Rs in the oral cavity, causing anti-inflammatory effects. So the mechanism seems to be similar. So if you have activation of these bitter receptors in different tissues, they cause protective responses. That's sort of a preliminary summary from our studies. So we'd like to acknowledge people in the lab. And Dr. Nisha Singh, she was a research associate. She is a research associate for the past 8 years in my lab, and then a PhD student and a BSc Dent student, DMD. So one of the good things working in a clinical faculty is you get a lot of DMD and MD students working in the lab. So he's a DMD student, and Firoz is again a PhD candidate. And so we have a microbiology expert whom we collaborate with in the lab, in the department, Kangmin Duan. John Hundren, he runs the CF lung transplant facility in McGill. So he supplies us with all the tissues we need, along with Bob, who is a research associate in John's lab. And the funding is from NSERC and CF Canada. They are the ones that are supporting our project on quantum sensing molecules and their role in CF, Children's Hospital Foundation too. Yeah, thank you.
Thank you, Prashant. Maybe we have time for a question or two, I guess. Yeah.
I really wish to know that you used model of T2R4 and T2R14. These are molecular models,
yes,
predicted models.
Yes, predicted and validated.
No, that's fine. How you predicted this model, I'm very much concerned about using a predicted model and then doing docking.
Yeah.
I'm really very, very concerned, and you are showing interactions at those means. How you really predicted the model?
Prediction, that's all. So validation and prediction, so we do mutational studies.
No. How you predicted the model?
Oh, these are iTaser. So we have
iTaser.
Yeah.
What was the template they took?
So iTaser takes multiple templates.
I know. What was your scores, Q scores? What was your Q score?
So
I'm curious to know.
We have 8 publications on that, if you want to look at those.
No, it's not the question of publication. I'm very much concerned a transmembrane protein.
And
you are putting a model and then docking something, a small molecule, and presenting molecular interactions. That's really, you are asking a tall order.
Yeah, okay, thank you.
There were mutational confirmations, right? Of what yeah.
Yeah. It's the immediate physiology is the triggering of a receptor response. Is there also some signaling back to the nervous system that could be important? I'm thinking specifically of super tasters who are much more profoundly sensitive to bitter, presumably through these receptors.
So yeah, that's one of the things. So we have a study going on looking at 80 kids so far. So the way we classify super tasters is based on one receptor, one of the T2Rs. That's known as T2R38. So there are 3 mutations in that, 3 different positions that affects G protein binding, right? So if we have those PAV phenotype, we classify them as super tasters. And those with PAV, AVI, tasters. So 25% of the population is super tasters, 50 is taster, 25 are non-tasters. So coming to that, studies have shown, not only our, other people's, that super tasters, they have more of a protective reflex. Protective reflexes, so because the thing is they don't sense it, like they're very selective in what they taste. Right? So they are more protective reflexes. Yeah.
Are there more questions? If not, I would like to ask you to stay put because there's going to be a video presented now that you don't want to miss. Thank you, Prashant.
Well, Govind, it's a real pleasure to be able to do this with you, to introduce you. and I'd like to introduce Professor Govind Khurana, who is at present at MIT in Boston. Govind, we're going to try at first just to go over the early part of your life and to develop that before perhaps we talk more deeply about science. Let's start at the beginning. Would you like to tell us something about your childhood in India?
Well, David, I like to first say just a word or two about the what I feel about being here. I think it's wonderful. It's a very special occasion for me to come back to England. You know that I spent actually many formative years in England studying just after the war, and it's always a great pleasure to come back to England, where I have so many friends. And of course this is a special occasion because essentially all of our former members of the group are going to be here. Now, to start with your question, I really grew up in a very, you could say, primitive, very simple village in India of about 100 inhabitants, and I would say that my father was essentially the only literate person, and I owe my ability at that time to learning to read and write to him. There was really no school to begin with, but he was, although a very humble servant of the civil servant, he was instrumental in getting a very small school going. There was one teacher, and usually there were very few boys that attended that school, but occasionally we had to really round up enough children because there was the school inspections, and if we did not show up adequate number, then the school was bound to be shut down. And after 5 years there, 4 to 5 years, mainly really being instructed by my father, after I went to another village, a larger place where there was junior high school, and later the family moved. But ultimately, I ended up at the University of Lahore, the Punjab University, where I went to, where I did the chemistry honors course, and actually that was just at the beginning of the war that I entered the chemistry course.
Now, would that be you would be exceptional, presumably, in going to university from your own village?
It would be. You're right. It would be because the literacy figure in India at that time and throughout India was no more than about 10%, and in the rural India, which was about 70 to 80%, this would be about 1%. So actually, I would say, in a way, looking back very proudly, that my brothers and I, and we had one sister, and we were the only eventually literate people out of that village. So I was very lucky to be able to eventually get to university.
So you studied chemistry?
I studied chemistry.
And
yes, chemistry course, the honors course, yes.
And that's perhaps where then the first glimmer of interest in chemistry started?
Well, this is something that, if I were to be honest, it is something that I should point out. I was also interested in English honors, and it was touch and go. I wouldn't say that I was one of these boys that sometimes who would say that, oh, I knew when I was this small that I wanted to do chemistry. The development of specific interest in chemistry came later, and but I was interested in chemistry at that time.
And so you did a first degree in India?
That's right. I did BSc honors, and then masters, and then the war came to an end, and after that I tried to go abroad, and fortunately through a stipend or a scholarship given by the government of India, it was possible for me to come to England.
What made you choose to come to England? And I know it was Liverpool you chose to go to. What was the reasoning for that?
Well, this was really sort of chance thing, because at the end of the war, first of all, I should say that the agency that was in charge of finding a place for us in the different universities or graduate schools was the High Commissioner's office in London. And it was really that office that found a place for me in Liverpool. But I think that they were trying all across the country at that time. I was lucky to be in Liverpool when I got there, because you really don't need very many people. You need just one strong influence. And I must say that in my case it was Roger Beer that influenced me greatly in chemistry, and also very importantly what I liked very much was the so to say the cultural aspects of life in this country.
So what type of chemistry then did you do with Roger Beer?
Well, it was very interesting. We began by first year doing natural product chemistry, something that Alexander Robertson, the then professor, had never done. But Roger, coming from Oxford, was interested in that. And we began working with alkaloids, which come from barks of trees that grow in New South Wales. And after a year, for various reasons, complicated reasons, we had to drop that. And then I went on to something that actually was even more interesting, because, and this was work on the melanin and its precursors. And it was very interesting to me recently that the work that I had done for my PhD on the synthesis and some initial work on polymerization of precursors of melanin, it comes in, brings in sort of sentiment to me, because, you know, I'm interested now in vision, to take a big jump, and we'll come back to this later, that in that one, the albinos have no melanin, and they therefore have quite a confusion in their retina, because the light that is not absorbed by the cone and the rod cells cannot be taken up by the melanin. But anyway, that's the kind of thing. So fortunately, I managed to get quite a broad experience: synthetic work, natural product isolation work, growth of bacteria, and of course the biology of melanins.
Stories are told that in those days you used to work harder than the average British postgraduate student. Stories are also told that sometimes you used to be locked in or out of that building. Are there instances you can recall of that?
Oh, yes. I remember that I was locked a number of times. There was a colonel who used to lock up at 6 o'clock, and I used to just somewhere, perhaps in some room, doing some experiment or operation, and would get late. It would get locked in. But I remember that also I used to get out by going to the back. There was perhaps the School of Tropical Medicine.
That's right.
In the old building.
Yes.
And there used to be a very high wall which had a glass embedded in this concrete on the top, and I had to be careful in not getting hurt and getting across that. Yeah. So that is really. But I did work. I'm not so sure how efficiently, but I worked. Yeah.
So that was then the start of your career, and you obtained the PhD degree at Liverpool University. Now, from there, you moved out of this country, I understand. You went on to Switzerland. So tell us a little bit about that.
I think what really was the main thing that I wanted to try to do, I knew I had to go back to India. I had come to this country with the support of the British Indian government at that time, and I was therefore under a bond to go back and accept a job if a job were to be offered to me, to go back for at least 5 years. So I wanted very much to get as much experience, broad experience as I could, and I at that time was very eager to learn to work in the German-speaking part of Europe, especially because in organic chemistry, the German science had actually been very strong. So, but of course Germany was out of the question, and I decided to try to go to Switzerland, and I managed to get there, but under rather unusual circumstances, because the government of India would not continue my scholarship, and I just went on some very modest savings that I had during my stay in Liverpool, and I spent a year in Switzerland. That was a very exciting time. It actually proved critical for my further work or direction, because not only I worked with a very exciting and inspiring man, chemist, Vladimir Prelog, but also had made new friendships, and one of these particularly influential in the sense that influenced my thought and work, was George Kenner. And we have a common bond in knowing George Kenner, and George Kenner became very good friends. And actually it was later, when I could not find a job in India on my return in 1949, it was actually George Kenner, a very loyal friend, who kept me in mind and was influential in getting me into the Cambridge laboratory, in Todd's laboratory at that time, which was a very lab working at the frontier of things in nucleic acid chemistry.
So just to go back a second, before you came to Cambridge, what type of work were you doing with Prelog in Switzerland?
Well, that also was alkaloids, natural products.
Mm-hmm.
So it was really very, Cambridge, Liverpool work, basically the work I had done in India before that, during the wartime, and also the work in Prelog's lab, was very different from what I was to do later on.
That's right.
Mm-hmm.
But it was George Kenner's influence, really, that brought
George
you
Kenner's very
to Cambridge.
clear, yes.
So at Cambridge, really, the work on phosphodiester's was really just starting in those days. And the Cambridge days, in fact, were very exciting days.
Yeah.
Tell us a little about it.
It's a bit more complicated, and something that I would say it was serendipity that got me into nucleic acids. In Switzerland, I came upon these reagents that chemists call carbodiamides, and I sort of rediscovered them and began to use them. And then I have to point out a rather esoteric or difficult, so to say, connection. But what happened that George Kenner was interested in peptides already. And I actually worked on peptides as well. But at the same time, I had this reagent which later proved useful, and also in peptide synthesis, as you know. And actually the phosphodiesters and nucleotides is an area that I worked on later, after I went to British Columbia.
Oh, I see. So what were you actually doing with carbodiamide when you were in Cambridge?
Well, what was already what one could see, and the experiment that I was very fortunate in doing was that carbodiamide could very rapidly and efficiently make pyrophosphate bonds. Now, Todd's laboratory actually had spent more than 10 years to make coenzymes, and these were important, biologically important compounds. But this carbodiamide reaction actually showed that you could do this faster in one step and really much more efficiently than any of the methods that had been discovered before.
Now, in Cambridge at that time were many men who, since that time, became very eminent scientists. Can you recall those people, one or two of them?
A lot of people that I had the good fortune to get to know. There was Alan Johnson, who became professor in Nottingham and then went to Sussex, and also had an MRC unit. Then there was Arthur Birch, who became at that time professor in Sydney, then came back to Manchester, and then after that went back to become, I think, a national professor in Canberra, I think. So those were two. Steve Angel was another one. Fergus MacDonald went to Canada. There was really a very exciting group of people that, of course, Basil Lithgow was leaving at that time. George Kenner himself then moved on to Liverpool. So it was really a very exciting group. There were people from all over the world, and I should also mention something that I will mention again, is that I got to know the first American that I got to know, and if a person that became a lifelong friend was Chuck Decker. Chuck Decker also coincided in Cambridge, and we actually worked in the same laboratory.
Oh, I see. I hadn't realised you'd met Chuck for the first time.
We met first time. Chuck Decker and I had very interesting, in a way, interaction, because I was trying to do peptides. There was nobody else that was doing peptides except Chuck Decker, who had worked for at least 3 years with Fruton, Joe Fruton, at Yale University at that time, which really shows how this, in a way, the excitement of science comes from the interactions with different people with different kind of background, but coming together, being particularly when there is a good place, and then they come together.
So just to continue the history a little bit, it was at this point then that you crossed the Atlantic.
Yeah.
You'd been in Cambridge, was it for 3 years or for longer than that?
I've been in Cambridge for, 3 years again, after having come back from India. And, well, actually at that time it was very uncertain as to where I would end up, because the situation in India wasn't very promising at that time. There was a possibility in Australia, actually. Victor Tricodius wanted me to go out there, but that wasn't. But then Gordon Shrum, a man who obviously began to mean a lot to me, he came looking for people, recruiting people to go to British Columbia. And so I accepted a job. It was a very simple job. There were really no facilities there, but Gordon Shrum said, but you would have all the freedom to do what you like to do. And that really gave me a great zest to just go out wherever I could and to get my own research started. And this is really how I went to British Columbia.
You know, there are all sorts, I'm sure you know, all sorts of stories told about people, and there was one story told about you at this time, that Shrum actually offered you this post, and you said yes, you would be delighted to go, and afterwards went out and asked people where Vancouver was. That is true.
I did not know where Vancouver was, but I would go there if you would offer me a job. That's really true, quite true. And I went and looked for a person who had come from Vancouver. I asked him. This was Ross Hall, but that's quite true. Yeah.
Okay. Can we stop at that point? Govind, I think it was 8 years that you spent in Canada. I know those years were very profitable years scientifically. Also, they were very happy ones for you. Sorry, did you want us to stop? Okay. Govind, I think it was 8 years that you spent in Canada, and I know those years were very profitable scientifically and also very happy ones. In fact, one of the things that you achieved in that time was the complete chemical synthesis of coenzyme A. Why don't you tell us something about the science and events that happened in Canada?
Well, David, I would modify perhaps this overall appraisal or statement about the 8 years a little bit, and something that we might come back to later, and that is that when you start out, it's a lot of hard work and a lot of persistence and many hours, and you are all immersed in what you want to try to get going. And so that was the case. Gordon Shrum was very encouraging, but obviously there's really not even pipettes around, and I used to work all day, and at night I used to wash my few dishes so that I could do experiment next day. And so this went on. And so the first year was really hard, but and Esther, my wife, worked actually also just assisted me in trying to be able to do these experiments. But after the first year, I had a postdoc, and particularly after 2 years, came a group that became actually famous all over the world, and every one of those members of that time is world famous. And then from '54, it really became very, very exciting. You could talk to people, you can discuss things and charter new ideas and experiments. But you're right that we were very fortunate that the overall effort of those 8 years was very, very rewarding. Also, you have to remember that we were very fortunate in starting work in an area which was really bursting forth right and left. There was the chemistry of phosphate esters, intermediary metabolism, nucleic acid precursors, protein precursors, and carbohydrates, and really everywhere you saw phosphate esters. So these were very much in demand. Secondly, the Watson-Crick structure for DNA had been, and also Brown and Todd structure for RNA had been worked out in 52 and 53. And here I was out there alone on a sort of desert island trying to struggle and develop methods for diesters, nucleic acids. But it really was, looking back, fortunately, a right kind of direction to go into. So we worked on methods of phosphate esters. The group worked on these, building really the basic knowledge about phosphate esters, about pyrophosphates, coenzyme syntheses, which culminated in coenzyme A synthesis, but also in the basic way to make di, tri, and oligonucleotide. All of this actually came out from during those eight years of British Columbian stay. So if I were to continue, then we expanded this to more biological lines later on in medicine when we moved there.
I see. But before we leave Canada, firstly, perhaps I should ask you to tell us a little bit more about one or two of those people. I mean, I know many of them are lifelong friends, and would you like to recall one or two of them?
British Columbia really was, for us, after the first year or two, but a wonderful and rewarding place, not only scientifically, but socially in every way. I'll show you later many pictures of that period, but in particular, Gordon Tanner and Mike Smith, who came actually 56. Gordon Tanner came in 54. John Moffat came in 54. Bob Chambers, the first postdoc that ever came to Canada, came in 54. And Ron Wright from Liverpool, from Stan Holker's lab, came in 54. And I think that these about five of us made this the hard core of this group. And then outside, there was Jack Campbell and other people at the campus that really helped us. And I could borrow things. I could not afford to have them, but I could borrow whatever I really wanted from these people. So altogether, and of course our children were born there, and Esther made lots of friendships. So I think altogether, socially, it was a kind of time when you make friendships that are lasting. I think just that phase in life, and also, of course, we had a lot of bonds between us in the laboratory.
So this is when you started to really to build your science family.
Yes.
Now, during your period in Canada, that was the time when you started to become well known as a scientist, and your work was recognized for what it was, and you were made several awards were given to you. Which of these achievements perhaps stands out in your mind?
Well, I think that there were awards and lectureships. I think the one that always means a lot to me of that period was the gold medal of the professional service. I forget, but it's a Canada-wide award, not given to just a scientist, but just it's really a professional service, like a civil service or any award across the country. And that meant a lot to me. And I want to emphasize another thing. At that time, it was very much in news whether I was going to stay in Canada or I was going to... Canadians are very self-conscious about it, losing people to scientists to the States. And they really were at that time afraid. They made every effort. I was offered the institute, independent institute. If I stayed in Canada, it was actually Leon Kerner, who definitely promised to me personally that he would have everything. This was a philanthropist actually from Czechoslovakia. But we had decided to leave anyway. But the thing that I wanted to say, and I have taken a long time saying it, is that this award was given after it had been well known in Canada that I was going to leave Canada. And so I really, appreciated that very much.
So that really takes us into the next part of the history. I think it was 1960
Yes.
that you made the move to Madison. Now I always think to up your roots, to make a move, there really has to be very strong purpose in it. And sometimes it takes courage to move from a place where you're being successful. So how did the move to Madison come about?
Well, I think that there was a lot of, difference of opinion among us. Some of us, such as Gordon and Moffat and so on, felt very strongly that we ought to develop strengthen our group in Canada, have this institute. I felt that, and as I did actually 10 years later, that it's a good thing occasionally to go to newer environment, and actually that's the reason why I left Madison 10 years later. But just for that reason, and I think that although it was very difficult, as you say, it took almost a year or even more to get things going again in Madison. I think that there were many things then we could do in Madison, particularly in the context of the work, exciting work in genetic code, more biochemistry that we could do in Madison that we could not have done in British Columbia.
Now, of course, Madison days were very exciting days, as it was once said, the golden era of molecular biology. Many exciting things happened there. Why don't you tell us one or two of the highlights as you see them?
Yeah, well, when you came, for example, David, in I think it was 63,
yeah.
We were just preparing to really, everybody was converging to that, if you remember.
That's right.
And we really got going in 64. And 64, 63, 64, 65, 66 was really the period when the results were coming out, and we had practically built all the background necessary for polynucleotide synthesis and use of these in elucidation genetic code and transfer RNA work by Thamraj Bhandari, Dieter Searle, and yourself, and also the genetic code that you did on the elucidation of the codons. And all that came about, and the 5 years were very, very exciting.
Mm-hmm.
And I must also say that it took some time, but after that we made the second switch, and looking back, that seems also, I can sort of be happy about that we made the decision that the next frontier was the DNA synthesis. And the next, Hans Weber, perhaps you remember, Hans Weber must have come just when you were leaving.
Yes.
And he really started a new wave, which was to culminate in the total synthesis of genes. So I think that Madison period actually was a unique period in the 10 years in my experience, because we, on the one hand, we solved a large problem, interesting problem, and then we went on to solve something that people did not really believe at that time was possible to do, the gene synthesis. And that came out in 1970.
And as a reward for that work, in fact, you did obtain the Nobel Prize. Again, there are sort of stories about it. Many stories say that most people in the lab knew you'd been awarded the Nobel Prize before you did. What were your feelings when you found that you had been awarded the Nobel Prize? And perhaps tell us how you did find out that you'd been awarded it.
Well, the award, as a matter of fact, it's to tell you now something that's not widely known. I got a call from some journalist very early in the morning, back 2 years before that the Nobel Committee is meeting and you are a strong candidate, and how would you feel if you got the Nobel Prize and so on. I said, well, I don't feel anything that I know about the Nobel Prize. And anyway, when it actually happened, I was, if you remember, I used to disappear into this motel, Karakal Inn, and where I could not be reached except by Esther. And actually it was there that Esther came out to tell me. So it was much later than the news had actually spread in Madison.
I see.
Yeah.
And what were your feelings?
Well, you feel, of course, happy, but I think in my case I also felt, I won't quite use Bernard Shaw's words about how he described his success, but I think there was a lot of excitement, but the sense of restraint that we really had to do new things, and it wasn't the thing that I wanted and then no more, that we wanted to do, really make new resolve to do new things. As a matter of fact, I remember that the group was very excited about it, and Hans van der Sande and Mark Crothers and Vittorio Scarmella and the whole group that was working on gene synthesis, they were even more excited to really, to start and push the next project.
Let's just move away from science for a short while. In your time, and perhaps through your success in science, you've also met many distinguished people and eminent people. For instance, I do know you've had an audience with the Pope. Would you like to tell us one or two things about these experiences and perhaps what has impressed you about meeting some of these people?
Well, perhaps I would. It is true that I am a member of the Scientific Academy, the Papal Academy, and a member of many other places. I think some of these somehow come. I must say I'm very proud to be the former member of the Royal Society. I think it meant always a lot to me to do that, although there are Soviet Academy and so on. But I don't know that it brings me particularly great satisfaction about that. What I think is more important is that when you get a prize like Nobel Prize, then there are lots of pressures on you. Pressures actually even more political. People expect you to solve all kinds of problems. People expect you to go to all kinds of places and address different kinds of meetings and so on. And I think the very, perhaps to me, the most significant thing that I accomplished, and maybe many people don't accomplish, perhaps I am being boastful about that, but the most important thing is to be very decisive and clear about what kind of path you want to take after Nobel Prize, because you can easily get waylaid. And if you want to stay with science, you've got to make sure that the answer to most of these things is consistently no. And that, I think, perhaps you would agree with me that that's what I've tried to do.
Oh, certainly, definitely, yes. Now, obviously you have a very agile mind, but at the same time you have a very agile body. I mean, you keep very fit, you're very strong. I can remember on many occasions we've been quite surprised by your own strength. Is this deliberate policy that you keep your body fit?
No, I don't. I'm not very actually disciplined in this regard. I'm not one of these joggers. I tried to start jogging many times, and after a few days give it up because I say, oh, well, I don't have time today, maybe I'll do it tomorrow. But I guess I do a little bit here and there. I do try to do a bit, but not, I guess I've been lucky, definitely. But in my building, I'm on the 5th floor. I don't use elevators. I just go up, run up, and down, or I may be going from one part of the campus to another, and I would just do this by walking briskly and so on. But I don't, I must say, I haven't made any deliberate. I swim. I like to swim, and I like to hike, and I like to go up mountains and so on. So, but these are, bits here and there. It's really not a sustained, regular effort. So I really have been just lucky.
I see. Yeah. Now, perhaps also with regard to physical welfare, you've managed to keep going all the time with keeping very unusual hours. Has there been any policy in this?
No, I don't know. I guess I like to do, like to work whenever I can work. I haven't always been able to do work efficiently. Sometimes, perhaps one should have a way that efficiency comes before really just punching the hours and so on. So I don't know that I've been very efficient.
Mhm.
Yeah, but I think I've tried to work. Yeah, tried to work very hard. Yes.
But always, the hours you've kept, it is because really that your life has been revolved around science.
Yeah.
Science has always come first.
Science has always come first.
So, if I can just take you away from science still a little bit more. So, what outside of science, perhaps, then, interests you, if there is anything at all, and perhaps in the same context? What do you do for relaxation?
Well, I think that I like to try to engage in things, pursuits that are not really very demanding, just things that are minor. For example, I like, as I said, I like to do things physical because there's so much work, the research work and thinking about the research group, the directions and so on, is really very hard work. So I like to escape by doing physical work, and for example, working in woods, even cutting down trees, and wintertime pruning trees, or just doing cross-country skiing, or just that kind of exercise. When I cannot work, for example, up in my cottage in British Columbia, in New Hampshire, I would just go out and just on snowshoes, I would spend 1 hour just walking through woods and up and down, or looking for different things and so on. So that kind of thing. Other than that, if I'm the other thing I do usually is I like music very much. So I would simply really listen to music.
And what type of music, Govind, do you think?
Mostly classical. Mostly classical music. Apart from I guess I like Sibelius very much, and Beethoven, and the other composers that we all like. But that, also the British, actually, Vaughan Williams, I like very much. Yeah.
And do you read at all outside of science?
I read now more than I used to. I usually read very few novels. So it's always something that is more, well, novels are very serious too, but basically general books, science books, biography, history of particularly science, but also history of other civilizations and so on. That really are the things that would interest me.
Now, you've been dedicated to science for so many years, and yet at the same time, I know your family have meant a great deal to you. How has your family managed to fit in with your dedication to science?
Well, I think you know that, David, that the science and family is the only two things. I had no other interest. You know that I have been, in a way, perhaps very fortunate. You have to make your choices, but fortunate in the sense that I did not have to do any lecturing. I never had to attend any committee work or go to Washington to sit on committees. And so it was really science and the family that came together. And the most important thing to say there is that my wife, Swiss wife, Esther, really has taken care of us all the way along in every way. I have been actually helped so much and given really encouragement, and also by her doing everything out of the banking, making tax returns, and making arrangements of all kinds, taking care of children and everything.
So actually, we all know, in fact, that Esther has given you marvelous support over the years. And not only you, but in fact, Esther has supported all of the group that you've had around you. I mean, we've all been able to rely on Esther for advice and encouragement from time to time. Yes, Esther has been really marvelous, I think, for all of us, hasn't she? So then the move came in 1970 to MIT. Tell us about the decision that you made there.
Yeah, that was a more difficult decision. in a way, because there was really no reason to move. I was in my 40s, 45 or so, but I had been given all the opportunity and laboratory and no claims on my time in Madison, University of Wisconsin. But the reason why I guess ultimately decided to move was that it might be a good thing to move. Different scientists that I would come into contact with, and perhaps there'd be new influences. That was really the only very abstract kind of thinking, but that's the only kind of influence or thinking that I had. I had a good laboratory in Madison. They built a new laboratory. Of course, MIT also built a new laboratory for me, or gave me a new laboratory. And in every way, I think there's no physical reason, except that it would be Boston, it would be an environment actually very rich, something that one has to work very hard to conserve one's time. But Harvard, MIT, and Tufts, and all these universities, and Harvard Medical School, where you can easily. So it was very different from Madison, really, or British Columbia, and I regret that in many ways, because the most important thing that you have is time and the ability to think without any interruptions, and you have to work harder in Boston area. But in general, I think it has been a very exciting move.
Mm-hmm. So in Boston, you've continued with nucleic acid work, but also you've opened up new horizons. I mean, in fact, you have done exactly what you said you would do. Tell us then about the work in MIT.
What happened when we left Madison, we had still to do a job on gene synthesis to find promoters, control elements, and then synthesize them and show that they work biologically. And this was done through the work of a lot of people in the lab, particularly in the final stages by Takao Sakiya and Mike Ryan. But then, by that time, I guess around when I was 50, I developed a very strong desire, a strong desire that I just could not get rid of, that even if I had to set aside several years or many years, that I had to develop something different, new interests, and I think that's what I try to do. Develop interest in membranes, and from membranes I became interested now what I find is really difficult but very challenging. For example, the sensory perception, the visual system, and before that I was interested in, and still interested in, is the light transduction by certain membrane proteins. And I think that this energy transduction in biology is a problem that hasn't really been touched very much, not at chemical or molecular biological level. And so I'm really, again, lucky, and I hope that the next 10, 20 years will be very exciting. I'm looking forward to working in these areas. But in addition to that, I should point out that what makes work in this area now very exciting is that we have, on the one hand, I should say that, as you remember, we were trying to merge disciplines already in the 60s. Mm-hmm. And I remain convinced that that any field that is moving, this tremendous this progress in the field comes by different tools and fields merging together. And I think that what I think now is that in these areas that I'm talking about, membranes, receptors, signaling in cells, and the responses they make, even oncogenes that people talk about, in all of this, we have to bring together, on the one hand, for example, nucleic acids, recombinant DNA work, with membrane methods, techniques, and how this way really the signals go from one part of the cell to the next. Mm-hmm. And that is the general area now.
Now, in order to mix together these many disciplines, obviously you must have to turn to many different people for their advice and guidance. Not just at MIT, but perhaps over the years, which people have you found have given you a lot of help, have given you guidance?
Yeah, we really have to. You're quite right. And we will discover as we go on. For example, the first need that I had about 5, 6 years ago, when we were working with this protein, light transducing protein that pumps protons, that we needed to know more about, and I am hopeless on that, about biophysics and physics, actually. So we collaborated with people, spectroscopy. You remember that Morton at the Liverpool campus had been a pioneer in visual research, and Wall also at that time. And they had worked out, for example, a flash photolysis, what goes on in the different intermediates in these proteins. And we have to do the same with visual research. And what we are trying to do is to, as the need arises, we collaborate with physicists, biophysicists, and of course we do biochemistry and recombinant DNA work. So cloning and structure-functional relationships we do in our own lab.
Now I know, Govind, in the Madison days, you used to go off to California to work with Arthur Kornberg.
Yeah.
Do you still do this from time to time, go off to other labs?
That's right. I think I should do that. And, but I must say that when I decided to work with membranes, proteins, I took mini sabbaticals, 2 to 3 week sabbatical, and I went to work with F. Ratter. And that really meant a lot to me, as working with Arthur Kornberg had meant a lot to me. A few weeks, but I think in a few weeks you get a sense of what kind of experiments, what kind of instruments, techniques people use, and that's what I did in the 70s. I have to do that again. I think that as soon as I feel that maybe for vision. As a matter of fact, it is true that I had a very strong desire last year to come to Cambridge to work with people here, with actually Sir Alan Hodgkin, who has been past president of the Royal Society. And this is electrophysiology. I think that there's really a great improvement, great new techniques in that area that we should learn. Particularly the new method, I don't know if you know, patch clamp. Patch clamp method is a very new method for doing really the new form of electrophysiology.
So we're sort of getting into this area. Would you like just to pick out one or two areas of science today which excite you, away from your own field?
I think that nucleic acids, molecular biology, are going to continue to be very frontier areas. For example, I think that activation of chromosomes and defining really all the controls in the expression of genes, that's really an open field. And then closer to home, in my area, I think that I mentioned before signaling, the receptors, insulin receptor or acetylcholine receptor or channels, conductance channels, opening and closing of these channels. I think this is a very important field. I think this is going to be very important. So basically what I'm saying is about really the new biological areas, and I'm aware of these more than perhaps other fields, biological areas that are coming into within the, so to say, scope of recombinant DNA and molecular biology.
Now, Govind, throughout your science career, not only have things developed well in the work itself, but also you developed unique relationships with your co-workers. For most of these people, the relationship has gone on over the years. And in fact, for many people, you really have now become a father figure, with Esther perhaps also being a mother figure. Why don't you tell us about these relationships, some of the people who you still have these strong friendships with?
Well, it's a very difficult, really, question. The first thing that I think we established in British Columbia, perhaps it had the germ for later work, was that nobody thought before that time that one could do any science there. And we tried to tell each other that we really did not have to be superhumans, that even if we just disciplined ourselves and worked hard, that we could do respectable science. And it really turned out that we did do respectable science in British Columbia. And I think as people came more and more many times people were not sure if they could do science, whether they should go to industry. You know, that's a phase when even after PhD, one may not be certain. I know many people that you and I won't mention names, but you know, and they were really uncertain whether they could make it or not. And I think that in all those cases, one thing was to try to understand really what they were thinking, and trying to, again, if they had this desire, to convince them that they could make it. And also the method, the way we went about, and I feel that the only thing I could contribute to was really an example. And if I worked hard and they saw how I worked and so on, I don't know what else could I do. And I think it turns out that we have really developed, this is obvious from this meeting we are having now, that a large, very large family of very excellent people, scientists that are very well known.
That's very true. Well, it all sounds as if it's been one big success story. But I do know there have been difficult times. Perhaps you'd want to recall just one of these times, and perhaps tell us what lessons it taught you when you were having a difficult time. Is there a lesson it taught you?
Well, I think the first thing that I have been lucky really, but I think the most difficult periods were the beginning. I think perhaps if I may go back, I think that the period going to Switzerland and not having a scholarship, actually, I've done my postdoctoral work without a postdoctoral fellowship, and it was not easy, but I enjoyed it. You never think as to whether you went to a restaurant with somebody or not. Later what you think, what influence did you have in that place, what did you do and learn. And later on, the possibility of not finding a job in India, because I was going around in streets in India when I went back, and there was no job. I could easily, I guess, have turned to, well, whatever job comes along, I'll take it. And I think it requires just at least a very strong desire that you really like to do research. And I think when I came from England and wanted second time, had this was 49 and then 52. And again, that wherever, so to say, destiny takes you, you'll go. But so that is perhaps the thing. I don't know what it is, but it is some motivation or desire. That is perhaps the most important ingredient, I think, for anyone to do. Of course, as I said before, British Columbia was very, very modest. There was really no facility. Gordon Shrum told me that I'm hiring a chemist and not a physicist, because the physicist is very expensive and the chemist needs only test tubes. And that's really what I had. If I had to borrow some apparatus, I had to take it. But slowly you work, and then things begin to happen. Something gives way, and things begin to happen. I'm sure that you and many people that have started from scratch have come to realize that you know, if you work away, some things happen that really you and I may not even have suspected it could happen, and then it gets easier and easier.
I was going to finish our interview by asking you a question which very nearly you've already answered, and that is, beside being a scientist, you're really quite a great philosopher.
Yeah.
What advice would you give to a young scientist starting off today?
Yeah, I think that that's right. I think that we just said that I mean, there are two kinds of people, perhaps. One that may say they already have such a strong motivation for science that that nothing else will satisfy that. And you know, a famous man, Auden, you know about Auden, W.H. Auden said that when you do that, when you know what your lot in life is, you have had your reward. Because that's the thing that you must do. So we're talking about the army of people that are not sure what they want to do, and I included. And I think for us, what we can do is really to develop a sense of motivation and a sense of values, really. You can ask for many kinds of, different kinds of life, and what you have to really ask yourself that this set of values is higher, or you'd rather be having this set of values than perhaps another set of values. I think that's the most important thing. After that, resources somehow, I think, sooner or later come around.
Well, Govind, thank you very much indeed.
Very good.
And may I say, on behalf of the Biochemical Society, that we do appreciate very much the time you've given us. And I'm sure that this video recording will be valued in time to come. Thank you very much indeed. Okay.
Thank you everyone. I thank Professor Nico. Now I'll think we have a short tea break. We'll start the next session at 4:50. Thank you.
वो बंद है